Okay, so if you recall yesterday's lesson, we covered what we call the central dogma. The central dogma is the concept that we start with DNA and we build proteins. There's a process that takes place along that way. We don't just go straight from DNA to proteins. There's several steps in between. And so we're going to look at that today. And I'm going to try to take this very slow with you so that way you can understand the steps of the process first. And then we'll get into the details of how that happens tomorrow. So I went ahead and I put a diagram of a cell on the board just to give you a visual to look at. Protein synthesis is this idea of us synthesizing or creating proteins. So that's what we're doing. We're creating proteins. When you look at this cell, notice that I labeled a couple of things for you. We went through all the organelles last week, and one thing that's really important for you to know is that there's a few organelles that are super important to be familiar with. One of those is, of course, the nucleus of the cell, because that's where you're going to find your DNA. The other is the ribosomes. I drew them a little bit larger than what they actually would be inside the cell, but that's just because I wanted you to be able to see them because they are literally protein factories, and I'll explain what that means in a minute. Protein synthesis is broken up into two big steps. And so let's talk about those two steps first. Transcription happens first. Translation happens second. Now, if you've ever seen the Amoeba Sisters video, or if you happen to end up watching the Amoeba Sisters video, she mentions in there the C and the S. C comes before S. That could be a little mnemonic device to help you remember that transcription comes before translation. Look at the two words, transcription. That means to write or something dealing with writing. I transcribe, that's writing. Translation, when you translate something, it can mean to read or to decipher, to code it, to understand it, okay? So in the first step, we're writing something. In the second step, it's being read. And that's where all of this comes in. Protein synthesis is difficult for students to understand. I'm going to try to keep it very, very simple for you to start. Once we start coding the information tomorrow, it might make a little bit more sense to you being able to see that coding process happen. So what takes place during protein synthesis, these two steps, there's something very important that's different about each of these steps, not just that one writes and one reads. Transcription happens when your DNA is ready to start producing proteins. A messenger RNA, and we showed that by putting a little M in front, will come in and it will build itself based off of the DNA basis. Okay, so let me repeat that again. Messenger RNA gets built based off of a DNA base. So what really happens here is, let's say we take this chromosome here, it gets uncoiled and a section of it, or a gene, remember that genes are sections of your DNA that code for traits. We might be coding for the enzyme, let's go back to lactose intolerance. This might be the gene that codes for lactase to help your body produce lactase. Okay, so this particular gene has been exposed. The messenger RNA is going to come in and read those codes and build a strand of RNA that is the complement to that DNA code. Okay, it's writing a new code as RNA. So this is similar to you writing a letter to a friend, which that doesn't happen a whole lot anymore. But let's say you're writing a letter to someone, or you're writing an email and you're sending it to someone, or you're texting a message. We'll use texting since that's probably the most common thing everybody does. You have something in your mind that you want to say. You text it, you code it out into words, into letters. And so that's what transcription does. This happens inside the nucleus. Now, if you think about it, it makes sense why this would happen inside the nucleus. It's safe in there, it's protected. The nucleus has its own membrane and can protect and care for this DNA. So as the messenger RNA is being written, it stays inside the nucleus. So that way it can be very safe and protected. You don't want that code to get messed up, because you're going to learn later, excuse me, you're going to learn later that sometimes when those codes get messed up, it can lead to genetic abnormalities. Sometimes that can be a good thing. Sometimes that's a really bad thing. So this is our first step of this process, transcription, writing the new code as RNA from the DNA. Okay, now what happens next? This little RNA moves itself out of the nucleus. It leaves. And it now puts itself into the cytoplasm. Remember, all of this is cytoplasm. All that jelly-like substance that's inside the cell. In the cytoplasm, you have the organelles, ribosomes. Those ribosomes are now the little factories, okay? So if we go back to letter writing, when you mail a letter, it has to go to the post offices. And the post offices then move your information around, whether it be letters or packages, okay? And so think of those ribosomes kind of like that. That code is being brought to the ribosomes. So the ribosomes can then read. We're reading the RNA code. Once we read the RNA code, we can then code for amino acids. Now, how does that work exactly? Well, I'm going to give you a really, really quick explanation, but we're going to focus on this a little bit more tomorrow. If you remember with DNA, you might have codes A, T, A, C, G, G, and I'll stop right there. I can base pair this with DNA. Now, if I base paired it with DNA, this would be T, A, T, G, C, C, right? I can also base pair it with RNA. Now, remember, when you base pair with RNA, A pairs with U, okay? So that was me base pairing my DNA. If I base pair with RNA, this will be U, A, U, G, C, C. My G's and my C's don't change. It's really only anywhere you have an A, you will place a U there, not a T. So this is my RNA code now. That's what just happened. We literally just transcribed that, okay? When this gets brought here, your ribosomes read it in sets of 3. 1, 2, 3, 1, 2, 3. And so it reads it in these little groupings, 3 bases at a time. This is actually called a codon. And so those codons can be read and can then be converted into an amino acid. Now, there's a lot of them, and I don't know of anyone that makes you memorize them until you get into some upper level biology classes. So we have a codon chart, which we're going to work with tomorrow and be more specific with that. But pretty much here's what takes place. That code gets written, then it gets read, and it's read in those sets of 3. So these 3 are one amino acid. These 3 are another amino acid. Depending on how long that DNA strand is from that gene will determine how many amino acids get built. And so literally the messenger RNA is red. And these cute little tRNAs, transfer RNAs, they come in and they match to that code and they click the amino acids in place. And this happens inside those ribosomes. And so you end up with this little amino acid chain that is built. Well, there's actually a special code to start and a special code to stop. We'll talk about those codes tomorrow. When they reach the stopping point, when that code comes out to stop building, this amino acid chain will actually start to fold. And every single amino acid chain will fold differently. It will fold up and it will form what we call a polypeptide, which is our fancy science name for protein. And that is the process of going from DNA to protein through these two steps, transcription and translation. Please understand there is a lot more to this process, a whole lot more. And tomorrow we'll actually do the coding part of transcription and translation so you can understand what exactly is happening and how the base pairs of DNA and RNA are involved in building amino acids and proteins. For now, it's really important for you to remember transcription occurs inside the nucleus, translation occurs within the cell's cytoplasm and works with the ribosomes. So let's get a few things together in terms of words. Let me find some space for us. You're going to see terms like messenger RNA, transfer RNA. This we talk about mostly inside the nucleus. This happens out here. M for messenger, T for transfer. They're both a type of RNA. You're also going to see those words transcription, translation. Okay. You also need to make sure you become familiar with the word polypeptide, amino acid. Remember, amino acids are just the building blocks of your polypeptides, which is your proteins. And then, of course, codon. We're going to talk about codons, the codon chart. Remember, all a codon is just a set of 3 RNA bases. That's all it is. Okay? It's a lot happening here. I strongly suggest you watch this video more than once, or you are welcome to go onto YouTube or go onto Khan Academy. Oh my goodness, there's so many places that have really, really great visuals, animations, pictures and things that have to do with the central dogma and protein synthesis. Some of it gets very, very detailed, some of it not so much, but this is a process that we're going to go over pretty quickly, but it is an important process for you to know. Okay? If you have any questions, make sure that you check with your teacher so that way those questions can be answered, because sometimes there's some misconceptions with this process. See you guys next time.